It's widely known that cancer cells continue to grow without slowing down or stopping. The general belief is that as cells age, this process could potentially block tumor growth and could eventually be used in cancer treatment. However, recent research in PLOS computational biology has recently demonstrated that although aging can occur suddenly in melanoma cells, it does not halt their growth, which is fueled by a small population of cancer stem cells. The study examined the connection between melanoma and cell aging. All cells undergo the process of decline and eventually stop duplicating once they reach maturity. The researchers followed the long-term changes in melanoma cell populations by closely observing the number of aging cells. They found that mature cells slowed their growth, with the majority undergoing the process after 3 months. However, the growth did not stop and eventually continued at its initial rate until the aging cells had nearly died off. The researchers then applied a mathematical model of the experimental data using a hypothesis involving the presence of stem cells. The idea behind this model is that a subgroup of cancer cells multiply continuously and are therefore unaffected by aging. These cancer stem cells are capable of producing larger populations of cancer cells, which are then able to replicate a certain number of times. The researchers conclude that despite a large percentage of cancer cells being vulnerable to aging, trying to replicate the process would likely be ineffective for treatment, as these cells are not critical for tumor growth. They do say, however, that the indirect evidence of cancer stem cells in melanoma could spark the creation of new strategies for the treatment of certain cancer types.